Good morning, everybody. Do. All right, let's get my screen up where we were last time. All right, we ended up here, which was talking about the cell-specific control of gene expression, right? And that is in many ways determined by what proteins are present in a cell, which is kind of essentially a kind of ancestral deal, not ancestral in evolutionary terms, but ancestral in terms of that cell's lineage, like what conditions experience. Good morning, Tony. Um, and the resulting changes in gene expression that make it that cell, right? So that's a big part of kind of how cells end up being different, right? It begins very much with the very first cell division, which is uh, sometimes asymmetrical, right? But not always. It depends on the organism, right? But anyway, so what we have left is of this PowerPoint is really talking about, oh, I'm sorry, no coffee yet. A few of the other cis-regulatory elements, and then moving on to some RNA stuff and then some protein stuff, essentially, right? So I want to get that done. And then we have some cool stuff to look at about using RNA silencing as a tool, and then some cool stuff on CRISPR-Cas9. All right, so it'd be good to finish that before the end of class 8, because this is the last one, right? So we kind of have to. And also because it's really neat stuff to know about, right? So it's kind of like the cool new school and cool cutting edge stuff. Okay, so one of the things about these enhancers, right? These you can see this kind of DNA bent round, right? So this isn't like a end flapping in the breeze. This is just to simplify the diagram. But DNA is kind of mobile, right? So it kind of folds around, and those proteins are able to interact with the transcriptional apparatus, even if that DNA is many, 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 many thousands of bases away, right? And so this poses actually a bit of a problem, right? Because DNA doesn't just bend in one direction, right? It can bend in other directions too. And so on a given chromosome, you'll have genes dotted along that chromosome, pointing in different directions and the like. But there will be a gene every, few tens of thousands of bases. So potentially these activators, right, can bind to enhancers and then activate other genes, right, which is not a good thing. And so really, oh, I got this. That's what I'm talking about, insulators, right? And so really you want to be able to insulate genes from each other, right? So that one gene is being activated by its enhancer or proteins bound to its enhancer here. Oops, put that back. But that enhancer or the proteins bound to it can't activate the promoter of a neighboring gene. Right, so essentially it kind of creates like these little walled gardens around genes. Now, if you wanted to coordinate gene expression, this would not be necessary, but many times you'll find genes which have very different roles, different purposes, or at least their products do, next to each other on a chromosome. And so you really want to avoid activating, inappropriately activating a gene because another one neighboring it is being activated. And that's what these insulators do. And typically what they do is they prevent DNA bending in a particular direction. right? So they kind of hold that DNA rigid so that it isn't able to flip over in the wrong direction and activate inappropriately this other gene. So silencers, on the other hand, okay, we have enhancers, right? Their role is to activate gene expression. We can also negatively regulate gene expression. We can silence genes, right? By essentially turning them off, right? And so these are also proteins, again, that can interfere with transcription somehow. So one thing that they can do is they can bind to these enhancers, right? So they can actually physically block these enhancers from being available to circulate and activated proteins. They can also bind to or near to the promoter, right? And so they actually interfere with the ability of RNA polymerase to even assemble, right? And so in that sense, it's kind of similar to the repressors that bind to the operators in bacterial gene expression. Similar, but not quite the same. Operators are really in between the promoter and the gene, whereas silencers are more typically bound to the promoter itself. And also just preventing the binding of transcriptional activator proteins. So if we go up here, and this is again another example of competitive binding, right? So typically you'll have these DNA sequences that are recognized by activators. And neighboring those, you'll also have other DNA sequences that are recognized by silencers. So the relative presence and concentration of those two different things kind of really determines the output in terms of transcription of that gene. And we've got an example here where, and this is actually by kind of a little bit more of a complex example where now the presence and absence of those different proteins determines the chromatin state of the gene to be transcribed, right? So this is an example of tadpoles turn into frogs. And essentially it's based on the presence and absence of different proteins, right? So in the presence of, so you can see this is a RXR as a nuclear hormone receptor, right? So it detects levels of a signal. And this thyroid response gene here, present in both cases. But it's what else happens to be present that determines whether this is a tadpole or if it's a frog, or going from one to the other. So in the absence of this T3 protein, right, this whole lot can assemble, and that recruits a HDAC. So that's a histone deacetylase, right? And if you remember from what we talked about chromatin, absence of acetyl groups equals really closed chromatin, right? And so by removing those acetyl groups, it shuts down this gene. No froggies. Now, when T3 is present, and this is most likely controlled by another process somewhere else, that enables this complex to form, which recruits a histone acetyltransferase, a HAT. Right, and so that acetylates these histones. Those histones kind of relax, the DNA comes off and is now available for transcription. Right, so this is an even kind of more complex example of how the presence and absence of different proteins promotes different outcomes in terms of transcription. Right, so these aren't just proteins. I mean, these are proteins that bind to DNA and these recruit these complexes to this particular location. but it's the other proteins which are assembling on those that form this complex that determine whether or not this gene is transcribed or not. So I think that's a pretty cool example. Okay, anybody got any questions before we move on to the RNA stuff? So this is all still about transcription. Okay. So moving on to RNA, right? So in terms of RNA, we have kind of three different things going on. Let me just bring up my chat. Where's my chat? Here we go. So with RNA, or rather mRNA, have three things. Oops. One, splicing. Two, RNA stability. And three, initiation of translation. Right, so those are the three different things related to RNA itself, right? Once it's been transcribed, that can affect the amount of protein being produced. Now, stability is obvious. If you don't have mRNA there, it's not going to get translated, right? So that's pretty straightforward. Similarly, if it's not able to be translated, you don't get any protein either, right? And again, that's fairly straightforward, although the mechanism by which that happens is not as one would expect, right? This is genetics. It's all complicated with lots of details. That's why people like me, love being geneticists, right? Because there's all this stuff to learn, right? And it's complex and it's fun and no one else knows what you're talking about, right? So, but one of the other real funky things, and again, this kind of goes back to the previous section when we were talking about splicing and RNA processing, right? So kind of hinted at like, well, what's the point of having introns, right? They take up a huge amount of space. It's another step that you have to go through to generate a functional mRNA. Like, why bother, right? And so actually the answer is not completely clear on that, to be honest. That's a bit of a rhetorical question in a sense. But one of the things that that does is it allows you to generate different proteins from the same piece of DNA and technically even from the same bit of mRNA, right? And so essentially, this is a good example of an insulin insulin receptor. Yeah, and so essentially, you want to have different affinities for insulin depending on how much of it is around. Right, so this is a little bit counterintuitive, but if you don't have much insulin there, you want a very high affinity for it so you're able to detect it, right? Now, if you have a lot of insulin around, you want to have a fairly low affinity because otherwise you will overwhelm that cell's ability to respond to that insulin. It'll basically get kind of overexcited. And that is actually a term. This is not like kind of a human analogy of like a small child that's been given a puppy. You can actually overexcite cells, and that's one way in which neurons die, actually, through overexcitation. But anyway, it's not a transgression, that's a diversion, tangent, whatever, right? But if you have the same protein, but you want to change its function, right, in a fairly fundamental way, one way that you can do that, other than post-translational modification, is to actually change what protein is produced, right? So in this example, we've got this is our pre-mRNA, right, our precursor, right? And there is also exons 1 to 8. They're just not included for clarity's sake, obviously, because we don't just start 9. But you have a 13 exon protein. So this is a big protein. Sorry, excuse me for a moment. I was just about to throw a placemat at my dog. And all of those introns get spliced, all of those exons get incorporated into the mature mRNA that's going to get translated. Now, when insulin levels drop, and you want to, or the cell needs to increase its ability to detect insulin, this exon 11 is removed, right? So this changes the mRNA that's produced. And so rather than splicing out the introns between 10, 11 and 11 and 12, this whole segment between 10 and 12 is removed, it's spliced out. Right, how that's actually managed, I do not actually know, because that would require a change in the splicing machinery. I do know that there is actually different forms of the spliceosome, which recognize different transcripts, but that's kind of the extent to which I know that. But somehow, magic maybe, this exon is removed from it was not incorporated into the functional transcript, and that actually changes the protein that's produced, right? And so in some way, that must change the structure of that protein such that it has a higher affinity for insulin, or is able to detect insulin at lower concentrations. And this isn't necessarily super common, but it does happen a lot, right? And actually the example that I showed you about the protein that I worked on, the gene that I worked on for my postdoc, one of them at least, that actually changes whether the it's not technically alternative splicing, but alternate promoters. But from the same DNA, you can have a protein which is exported into the extracellular space and one which is retained within the cytoplasm, right? And so this is kind of like the point that really these alternate splicing and alternate promoters get at, is that you can have the same bit of information, the same bit of DNA, but you get very different proteins in terms of their functional location from that, right? Which is actually pretty neat if you think about it, right? Because you're not changing what genes express, you're just changing which bit of it is expressed or which bit of it is turned into protein. Now, now we're getting on to this, right? So this is about stability and initiation of translation. This is actually a very fundamental ancestral process, right, which all eukaryotes have. And essentially, there's two different aspects to this. One is a cellular defense mechanism against double-stranded RNA viruses. The other is a means of modifying RNA stability in a transcript-specific manner. Right, so essentially, if you think about transcription, once that mRNA transcript has been produced and exported into cytoplasm, it will get turned into protein over and over and over and over and over again. And so if you only want the production of that protein to occur within a specific time period or in response to a specific event, such as a signal from the outside the cell, you want to get rid of, not only do you not want to turn off transcription, that's all well and good, but what about all the mRNA that's still floating around from the activation of that gene, right? You're going to need to get rid of it unless you want protein to be produced, indefinitely, which almost certainly you wouldn't, right? Particularly if that gene is expressed in response to something like a change in diet or a particular nutrient or whatever, right? Some specific cellular cue. And so you want to turn that gene off, and you want to get rid of that mRNA. And the way that we do that is that we target specifically that transcript. So if you remember me talking about the 3' UTR, the untranslated region in mRNAs and eukaryotic mRNAs, that is typically the region which is used to control mRNA stability, right? So the 5' UTR is in many senses to do with translation, right? That's where the small ribosomal subunit binds. or the other gum, right? The 3' UTR is a lot about control of translation and transcript stability. And so this occurs via a really funky mechanism. In fact, the discovery of this super funky mechanism garnered 2 C. elegans researchers a Nobel Prize. Andrew Fire and Craig Mellon, I think his name is. Gives terrible presentations. I watched him speak once at a conference. It was absolutely horrible. But anyway, so it just goes to show that even if you get a Nobel Prize, doesn't mean you're a very good talker. This gives me some comfort, right? Because I'm never going to get one. Anyway, so they discovered this by accident, which is actually really cool because I love, discoveries that happen by accident because they're often the most interesting ones. And so basically they discovered this mechanism by which if you introduced a bit of RNA to a cell and that RNA was specific to a gene, that RNA got used as a kind of a scanner or a code to find that little fragment that you introduced, to find the transcript that you're targeting, and it gets destroyed. Now, what they found, which was even weirder, is it doesn't matter whether that small piece of RNA is complementary, i.e. it will bind directly to the mRNA, or the reverse, right, the antisense. And so what they actually accidentally discovered is it's all about double-stranded RNA. And so if you introduce a bit of double-stranded RNA to a cell, that cell recognizes it as, oh shit, we've got something here that we shouldn't. And it goes through a whole series of steps to a point where that bit of RNA is used as a barcode scanner to find the target mRNA. And so cells actually use this themselves to control transcription and translation, or not transcription, transcript stability. And so essentially what happens is that you have these small pieces of mRNA, not RNA, sorry, non-coding RNA produced. Right, so these are expressed from genes which code RNAs, but those RNAs are not translated. So these are called non-coding RNAs. If I get my chat box up. Oh, sorry, Tanya, I didn't have my chat box up. Let me just type this out and I'll get to your question. So one key point is that these non-coding RNAs, let's stop doing that, are produced in the same way that mRNAs are produced, right? They're from genes, they're under control of promoters and regulatory elements, and they're transcribed like other mRNAs or other RNAs are, including things like tRNAs and ribosomal RNAs and stuff like that. So these include, obviously, rRNAs, tRNAs. We've got small interfering RNAs. We've got microRNAs, not to be confused with mRNAs. We've got Pee-wee RNAs. Don't know what those are. I'm always curious because it seems such a silly name. And so on, right? Because they're just more than I know of, to be honest. All right, let me get to Tanya's question. I'll move on. That's an interesting question, Tanya, about the role of introns. I mean, this kind of gets into my kind of fuzzy hand waving response mode, to be honest, because I don't actually have an answer to that. I don't know how specific introns are recognized so that an exon or two is spliced out. My guess would that there'd be a specific sequence. So remember we talked about the splice acceptor sites that are just inside at either end of the intron that are recognized by the spliceosome. My guess would be that those splice acceptor sites there are additional ones, let's say, or there's additional information that allow those to be recognized by a different spliceosome. And so by controlling the presence of the components in that different spliceosome, you're able to then control whether or not just the introns get spliced out, or if an exon gets spliced out with it. So there's not necessarily any information in the intron that kind of says, hey, this exon should be here or not. I think it's more that the cell can control what spliceosome is used. And because different spliceosomes, and I know there are different ones, and they have different, they recognize slightly different sequences in introns. By controlling that, then you control how those introns are spliced out. So I think it's by controlling the machinery that does the splicing, not by any signal necessarily coming from the intron itself. But those introns have to be slightly different in order to be recognized by the different machinery to give you different mRNAs as a result. Yeah, that's a little bit arm waving and, but that's I think a fairly reasonable educated guess based on what I do know. And I don't know, that is like a whole nother field which I just don't really super understand. Yes, Levia, that's completely correct. So before splicing, it's precursor mRNA. It's not a mature mRNA transcript. And so really precursor mRNA is anything before the mature mRNA is finished, right? So it also includes mRNA that isn't capped. It's mRNA that doesn't have the 3' poly A tail, and also mRNA that isn't spliced. So all of those three things have to be done for it to be considered a mature mRNA transcript. But yeah, so before splicing, it's most definitely pre-mRNA. Cool. Okay, so getting back to this, right? So we've got these small RNA transcripts produced, and these actually form secondary structures. They form this double-stranded RNA, right? And they can typically do so by forming stem loop shapes. Is that with you, dog? Yeah, doing something funky, right? And so this is the key, right? The presence of this double-stranded RNA is the trigger that activates this whole machinery. And so essentially the key point to remember, and this is key to both how it works endogenously in the normal cells, but also how we can use it as a tool. Part of this sequence recognizes or is complementary to its target mRNA. So this non-coding RNA will have a small part of it which matches either precisely or somewhat precisely the 3' UTR of its target mRNA. And so when that double-stranded RNA is recognized, or it goes, woohoo, I'm here, right? And essentially what happens is an enzyme called dicer, which is specific, it's an RNA specific to double-stranded RNA. It recognizes that double-stranded RNA and it cuts it into small bits. The size of which actually differs for different non-coding RNAs, but it's not super important. And basically it's between 20 and 25 or thereabouts nucleotides, right? So this larger piece, and it can be, in a few hundred base pairs, right, gets cut into smaller chunks, right? That's the first step, right? So it goes from this to these small little chunks of double-stranded RNA. Now, those are recognized by another complex, which is the RNA-induced silencing complex, or RISC. And what this does, and actually has a whole bunch of proteins, including this one called Argonaut for some reason. Don't know why it's called that. From presumably someone like watching old fantasy films. Probably no one's ever watched Jason and the Argonauts. It's a terrible, terrible film. And the CGI is just, it's not even C. It's just GI, I guess. Is actually pretty cringeworthy, but fun to watch if you want to giggle, have a few drinks and watch that. Anyway, this complex assembles around these small fragments, and its job is to do two things. One, it chews up one of those strands, right? So for that RNA to recognize, this target RNA, or this barcode RNA, I guess is probably the better word. For it to be able to recognize its target, it has to be single-stranded. Otherwise, you don't get the hydrogen bonding, right? And so the first step is that RISC complex chews up one of the strands. And now it has a kind of a barcode, and it is literally going to hold that, and it's going to go around going, nope, 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 to every single mRNA it can find. Right? It's literally just wandering around your nucleus or nuclei, trying to find a match. And it's going, nope, 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 nope. Oh, and it finds a match. Now, what it does depends on how good of a match it is. And this is where we get that distinction between transcript stability and control of translation, right? The balance between the two. If it is a perfect match, or nearly perfect, then the Argonaut cuts the mRNA strand. And as you know from mRNA processing, the ends of mRNA are protected to prevent degradation by RNases. So a free 3' or 5' end of mRNA that has nothing attached to it is like yummy sauce for the RNases in your cells. And they will recognize it and they will chew it up. And so that mRNA transcript will disappear. It will be literally chewed up. It'll be degraded. That's if this match to the target is perfect or nearly perfect. Now, if the match isn't quite perfect, right, and there's kind of a range of how good of a match it needs to be for something to happen, and how. Sorry. Hang on. Oh, she stopped doing it. My wife was trying to distract me while I was teaching. It worked. Yeah, she's all super happy now. I'll get my own back. Don't worry. Anyway, my dog's come to give me some comfort. Look. Yeah, there we go. You'd have been nice to me, aren't you, doggo? Anyway, so there's a range between which, under which nothing happens, right? Because it doesn't recognize the target, and over which the target gets degraded. And kind of between that, and I think it's around 3 to 4 mismatches, give or take, right? If most of the guide matches, but not all of it, I don't know, say like 80% or better, then instead what happens is that the binding of that RISC complex prevents the mRNA from flipping round. If you remember, one of the roles of the poly-A binding proteins is that the mRNA flips round, and the poly-A binding proteins help the ribosome assemble on the 5' cap. And so by preventing that mRNA from doing that, it's like, I can't do it, right? I can't bend because this RISC complex is present, prevents translation. And so depending on the degree of matching, whether it's perfect or, yeah, mostly perfect, if such thing can exist, determines whether or not that mRNA is degraded, right, which is terminal, right? No mRNA, no translation. Or if translation is inhibited. And I don't actually know whether this is transitory or not. I don't know. I don't know if it, once it's bound, it's always there. And so that just shuts down translation completely from that transcript, or if that's something that can stop. I don't actually know. But this gives us an extra wrinkle, right? An extra way of controlling gene expression. And this was actually discovered as a means of controlling gene expression in C. elegans again, because it is the world's best model organism, in my opinion. She's doing it again. And what they actually found is a non-coding RNA that controls molting. So you probably know from insects, right? They go through different kind of larval stages and they shed their cuticle as they get bigger. Worms do the same, right? Or nematodes at least, right? And so it's a periodic. They'll grow for a bit and they kind of like shuck off their cuticle, and then they grow for a bit more and they shuck off their cuticle, and so on and so forth. And so the actual process of molting, which is what that's called, is a cyclical one, right? Doesn't happen, happens. Doesn't happen, happens. Doesn't happen, happens, right? And so that has to be controlled by some kind of periodic signal. And it turns out that that periodic signal is actually a non-coding RNA. And so its expression peaks as molting occurs and then drops to nothing, and peaks and drops to nothing. And so its job is to essentially shut down normal gene expression in favor of a molting specific gene expression. So that's how it's discovered. And really this is one of the probably the great unknowns of eukaryotic genetics, right? These genes are very hard to find because they don't produce protein, right? So A, they won't produce, a protein that you can find. And B, they don't actually have the same kind of markers, I guess, right? They don't have a start codon to look for or stop codon, right, in the DNA. And so we don't actually know how many of these non-coding genes we have. And maybe a lot. And certainly a lot of what we thought was junk DNA actually codes for these things. Right, so however complex we think eukaryotic gene expression is almost certainly far more than that. Anyway, okay, so now we've got to move on to proteins, and Ryan, this is the last step on this PowerPoint. And so this is actually something which tends to surprise students, right? And people that haven't spent a lot of time working on this subject, right? There's the actual protein that you produce from an mRNA through translation is rarely the final product. And proteins themselves are activated and deactivated, not just in a binary like you're on and you stay on, or you're on and we turn you off like forever, but in a dynamic process, right? So there are a lot of proteins which are continuously being flipped between an on and an off state, right? And whether they're on or off determines other outcomes. So those proteins themselves often turn on other proteins, which turn on other proteins, right? And so you get this huge web of control which has nothing to do with the control of transcription or translation. So once these proteins are produced, they also are controlled, their activity, right? So a large part of what goes on in cells, both every day and during development, in many ways has nothing to do with the transcription of the proteins that control that process. It's all about whether those proteins are turned on or not whether they're present or absent, but whether they're active or inactive. Right. And so this is where it starts to get really kind of mind bending in a sense, because it's almost kind of how I feel whenever I read astronomy magazines, right? And they talk about parsecs and, the bending of light through bending of space time, 4 dimensions and things like that. It's like, makes my head hurt. I have to go lie down for a little bit in a dark room. And this is really kind of like that, right? It almost becomes so complex. It's almost understandable, right? How much is going on. So quite often in terms of science, we break it down to fairly kind of narrow views to try and understand it better, right? Because it's just a very hard thing to understand. And so, but this is actually one of the more interesting parts, in my opinion, right? Typically, people are very interested in the control of transcription, i.e., what goes on up here, right? But there's a whole nother world of control and intricacy and complexity, which happens all the way once you actually have a protein produced, right? And so these modifications are many and varied, right? And then typically most, not necessarily all, but almost all eukaryotic proteins are modified in one way or another at one point or another. And these modifications are very varied, right? And so you can add functional groups, right? And so these are quite often phosphate groups. In fact, phosphorylation is the most common type of modification, right? So I hate typing phosphorylation. It's almost as bad as trying to type out phosphatidylcholine over and over again, like I had to do for a paper. Absolutely sucks, makes my hand hurt. So that's addition of let's move this over here, phosphate group. So that's a PO4 by a protein called kinase. And so this is kind of like with histone acetylation. You have one protein or type of protein that adds them and one that removes them. And this is the same for phosphate groups. And so the opposite would be a phosphatase. And the balance, and some proteins can be phosphorylated at multiple points. The balance of the various combinations, it's not just a one, add one and it goes from one to the other, but it's a combination sometimes of, is it phosphorylated here but not here, equals one type of activity. And the opposite might be inactivity or a different type of activity, right? So it's really complex. And also, and this is where it gets even funkier, can have kinase cascades, right? And so basically the idea here is that once a kinase is activated, it can activate the next kinase by phosphorylation, and so on, right? And so you can have series. There's actually one that I've worked on that has 3 kinases. It's a map kinase, a map kinase kinase, and a map kinase kinase kinase, because the people that called them that weren't very imaginative, right? So you can have this whole kind of cascade. Also, we have attachment of sugars, right? So many sugars are glycosylated. Glyco being the sugar, silation is addition of, essentially. And these proteins are quite typically on the outside of a cell, right? So they're localized to the cell membrane, and those glycosyl groups are external facing, right? So in many ways, cells are recognized. It's like, oh, that's one type of cell, by sugar groups. And actually, I think at least part, I've got to be careful. At least part of your blood type, I think it's the rhesus factor. Yeah, I think it's your rhesus factor. Maybe even the ABO, but I'm not 100% sure about that. Is down to how those external facing proteins are glycosylated. And quite often those sugar groups are recognized by your immune system, right? So they're one of the main ways in which bacteria are recognized as a bad thing if they happen to be floating around in your blood, is by those sugar groups. And another major modification is cleavage, right? So those proteins are cut. And quite often you'll have a protein which is inactive until it is cleaved, and then it becomes active. And the control of that cleavage is the way that you control whether that protein does its job or not. And so I've dug up a couple of cool examples, because I think this is fun stuff. It's worth illustrating, right, rather than just telling you stuff. So this is a great example of phosphorylation and how it can activate or inactivate a protein. And so this is C. elegans. This is a parasite of humans called Strongyloides stercoralis. And this work was actually done by a friend of mine called Michelle Castellito I think she's at UCLA now. Anyway, so DAF16 is a transcription factor. So specific type called a forkhead transcription factor, right? And it's involved in turning on genes in response to insulin signaling. And so given that it's a transcription factor, it can only be active if it's in the nucleus, right? And so the way in which DAF16 activity is controlled is by controlling where it is. And so it can be either in the cytoplasm. So this is a GFP worm. And so this is a cell. You can see that small kind of bright green dot with a little dark bit in the middle of it. That's the nucleus of that cell. I don't actually know what that cell is. Could be many, right? And the slightly fuzzier green is that protein in the cytoplasm, right? And so its relative location determines the relative output in terms of transcription of the genes that it activates. And so in the absence of signaling, right, that protein is phosphorylated, has a phosphate group added to it, and that pulls it into the cytoplasm. So it removes it from the nucleus, and it's now unable to turn on transcription. Now, when signal is received, those phosphate groups are removed by phosphatases, and now it translocates back into the nucleus, which you can see here. This is kind of a much two bigger cells, and it's now able to activate transcription. And you can actually study that by making choice mutations which change those residues which can be phosphorylated. So 3 types of amino acids can be phosphorylated. So it's serine, threonine, and tyrosine residues. So obviously proteins quite often have more than one of those. So that kind of shows you how much you can do potentially, right, in terms of phosphorylation. And so if you actually change those residues in this protein, you change it between ones which can never be phosphorylated, you get this result. And so it's always in the nucleus because it can't be phosphorylated and removed. And there are actually residues which kind of mimic what a phosphorylated protein looks like. I'm not entirely sure how. But if you change one of those residues to that instead, now it's always going to be retained in the cytoplasm. And that's what this kind of experiment was actually all about. Okay, so that's phosphorylation. It doesn't necessarily have to be about localization. It could just be about activity too. But this is a neat example because activity is tied to location, right? And so by just changing where the protein is, you change essentially whether it's active or not. And the next example I want to give, which is a really cool one, I really like this. This is all about not bleeding to death, right? And so when you have a cut or an injury or something like that, you want to seal that hole up as quickly as possible for 2 reasons. One, you don't want to leak all your blood out because that's, useful. Without it, you'll die. Actually, you don't have to lose very much blood before you get into serious risk of death. I think it's like 3 out of 9 pints, something like that. Bad stuff starts happening. And also, this is a hole in you, right? And this is a way in which stuff that shouldn't be in you can get inside you, such as bacteria and fungi and various other nasties, right? And so you want to seal that hole as quickly as possible. And so what happens in a normal individual, obviously, if you're a hemophiliac, this doesn't happen, which is why hemophilia is so dangerous. In a normal individual, once that blood vessel gets broken, that triggers the release of what are called clotting factors. And those do 2 things. One, they cause the blood vessel to constrict, right, which slows down blood flow and reduces blood loss. And 2, it activates platelets. And so platelets are just proteins that kind of hang around in your blood and then are able to stick blood vessels together. And then the second thing that happens after that is this mesh of fibrin. And so fibrin is basically like a net or a mesh that kind of holds that clot together, right? So the first thing is that you plug the hole and then you make that kind of essentially permanent, right? And this happens through a cascade of events, right? The first is the production of these clotting factors. And these clotting factors, actually one of them, can't remember, 8, I think, is what is deficient in hemophiliacs, right? So they don't produce one of the clotting factors. And one of those is an enzyme which cuts this circulating protein called prothrombin, right? And so in normal blood, no injuries whatsoever, you have prothrombin and fibrinogen and inactive platelets floating around in your blood. They're always there, right? Ready and waiting, but not doing anything, which is good because you don't want blood clots. Like that's a terrible thing, actually. That's what leads to strokes and heart attacks and stuff like that, right? So these things should be completely off when everything is normal. But as soon as you get a damaged blood vessel, a broken blood vessel, the lining is damaged, those clotting factors are produced, and one of those cuts or cleaves this protein from prothrombin to thrombin. And this is an enzyme which then cleaves a second protein, fibrinogen, and turns it into fibrin. And essentially what happens is fibrin sticks to itself. Is fibrinogen? It doesn't. So it just floats around as a single monomer. When it gets cleaved, it sticks to itself and it forms a mesh, right? And so this multi-step process is actually quite common where you want to have very tight control of a particular process, right? So multiple events must happen for this to work. And also this is a way of expanding the response so it happens very quickly, right? This is a cascade of events. And so what this looks like at a molecular level, right, and this is a kind of a space-filling model of prothrombin, is that two of the clotting factors basically cut the protein, right? So they recognize a specific shape of protein, and they will cleave it at a very specific peptide bond. And when they do, they release thrombin, and thrombin is an enzyme, right? So it's inactive in this form. It doesn't do anything. It has to be on its own for it to be active. And as soon as it is on its own, its target is fibrinogen, right? And so fibrin actually it looks way funkier than this. Let's have a look. Right, so it's actually it's a really weird looking protein, right? It consists of, I think, 6 different strands, polypeptides, and they basically have these balls on the end and this region in the middle, right? And this region in the middle is the key one for it sticking together, right? And so essentially what thrombin does is it just chops off little bits around that center part. Right, these A's and B's here. And by doing so, fibrin now sticks to each other, right? It basically has that kind of interaction that can now occur, and it forms this large kind of multi-unit mesh. And what that does is it stabilizes the red blood cell and platelet blockage, and it forms a permanent, well, semi-permanent seal of the damaged blood vessel, which can now return to its normal size, and blood can flow and everything's back to normal again. Because you also want this to happen pretty quickly, because you don't want to cut off blood flow to really any part of your body, but some parts in particular, like your brain, because that oxygen in that blood is very, very important. So this is a pretty exquisitely controlled process, right? Which you want to occur very quickly when you need it, and to never occur when you don't. And so the control of this is actually, I think, pretty cool. And it's also a great example of protein cleavage resulting in active protein. Okay, so we've only got 15 minutes left, which is sad. I'm sure you're all shedding a silent tear. probably that will wait until after the exam, I imagine. Um, but I want to show you just some cool stuff, right? This is in the transgenic. Oh, look at that. We've got a fat mouse. This is really cool. This is a leptin knockout mouse. Basically never knows that it's full. Anyway, so this is in the transgenic PowerPoint. It's actually a new one which I uploaded last night to have this information in it. And I just wanted to show you some neat stuff. And so one of those is this. Isn't that an amazing picture? This is an alligator embryo, which is expressing different fluorophores, right? Different fluorescently labeled proteins. And the different colors are, I think, from memory, green is nerves, which you can see here. Blue is muscle, right? So you can actually see the developing brain here. And these are, which I always think are super beautiful, these are called somites. So these are the developing blocks of tissue which will become your vertebra. And so I think that's such a breathtaking picture. It really is. This was done by someone at Yale, basically trying to compare alligators and chickens. That's kind of the aim of that particular project, but produced a really pretty picture as a result. Another thing that I want to just show you is, so we talked about, where's the other one? This process, right, which is all about transcript stability and initiation of translation. Well, what you can do is actually make your own, and thankfully someone did that for us. But you can actually make libraries of bacteria that express double-stranded RNA to every single gene in an organism. And so in C. elegans, you can buy, if you've got, how much is it now? I think it's $15,000, which isn't much, price for a nice secondhand car. You can buy yourself a library of every single gene in C. elegans, right? And so in C. elegans, the cool thing is you just have to feed those worms that bacteria. So you make an agar plate, which you're familiar with for growing bacteria on. You put the bacteria on it that expresses the double-stranded RNA. The worms come along, suck it up, and somehow, partly through magic again, which is basically means we don't fully understand the mechanism, that double-stranded RNA is taken up by the worms and it knocks down gene expression of that gene. And so you can do all kinds of funky experiments. So this is, and worms are neat because lots of it's really easily visible, right? So this is a normal worm, N2 wild type. And if you knock down this gene, whatever this does, or this one, you get this really nasty cuticle phenotype, right? And so this is basically the old cuticle from a previous molt. Well, actually, this is the cuticle lifting off. This is the old cuticle from a previous molt. And so these worms kind of basically choke themselves, right? Because they can't get out of this cuticle. And so you can do that by either getting a mutant, right? So this is a mutant allele, right? Through a standard forward genetic screen, doing mutagenesis. Or you could simply pick out one of the bacteria from your library, feed it to the worms, and you get a very similar phenotype, right? And so you can actually modify the phenotype of the worm without changing its DNA, right? So you can, rather than going through the labor and the randomness of mutagenesis, instead you can go, I want to knock down that particular gene, feed it to them, and you get a very similar kind of phenotype. And so that gives you a lot more flexibility because A, it's a lot easier to do, and B, as well, some of these genes are actually required for function. So you'll never get these genes in a mutagenesis screen because the worms will never survive. Right, so any worms that are homozygous recessive for that mutation die. They're lethal mutations. And so RNAi actually allows you to conditionally knock down gene expression and look at the results without having to deal with any kind of side effects of knocking that gene out. And you can do that in Drosophila too. They're much uglier, obviously, because they're flies. But in flies, instead, what you do is you actually introduce the genes for making that double-stranded RNA, and then you add something to their diet which promotes the induction of that double-stranded RNA. So rather than doing it from the outside in like C. elegans, you actually put those genes into these flies and then turn them on. And so these are just some examples, these 3 different genes, and essentially what they're showing you is that you can get the same phenotype from RNAi as you can from a mutant. Right, so just by turning that expression of that double-stranded RNA on, you now get a small white eye rather than a large red eye. Or overbanding on the abdomen, similar to what you see in males, but this is a female. Or these, I don't know what that is, actually, some marking on the fly's head. Right, so you can actually do this also conditionally, right? So you can say, if I knock this out at this particular point of development, what effect does reducing transcription or gene expression really have on that fly at that particular point? So you can do a lot of real kind of funky sort of experiments and real fine control over what you're doing, which is super neat. And then finally, and this, to be honest, 8 minutes is not going to do it truly justice, we have the latest hotness, right? So RNAi has been a utterly transformative technique. It really has. It's kind of hard to, almost on the scale of PCR, I would say. But it really has almost nothing on CRISPR-Cas9. And so the problem with working with most organisms is generating these mutants is random, right? You basically screw them up either by gamma radiation or addition of nasty chemicals like EMS or whatever you would want to use. And then you screen all of those screwed up animals for the phenotype that you want. So you have no control over where those mutations occur or what type of mutations occur. It's random. And some of them you'll never get mutations in because those mutations will be lethal. Now with CRISPR-Cas9, you can design those mutations yourself. And so if you want to say, I want to change that amino acid to that amino acid in this particular place, you can now do that. And it's really, really hard to overstate how big of a deal that is. And so essentially now, if you want to kind of take the apocalyptic kind of viewpoint, now if we're able to associate or find associations between a particular protein sequence and intelligence, now there's actually nothing other than our ethics from stopping us changing those sequences in humans. The technology is there. It's not that we can't do it. It's that we as a community are choosing not to do it. Right. And in fact, there are people that have chosen to do it. There's a case you probably heard of this in China, of a scientist who introduced a mutation into a cell surface receptor called CCR5, which is used by the HIV virus to infect T cells. And so obviously without those children's consent, because they weren't even children then, they were embryos, he modified their DNA using CRISPR-Cas9. And so now those children have that change. They were are they twins? Can't remember. Can't actually remember all the details. And their offspring will also have that change. And we don't actually truly know what effect that change will have. Right? Obviously now they're much less risk of getting AIDS, but there also might be a much greater risk of getting other viral diseases instead. And so this is actually a kind of acquired immunity for bacteria. So this is basically bacteria, different types of bacteria use this for recognizing when they've been infected by a virus. And so they keep a little bit of that viral DNA and they stick it into their own genome in a cassette. And they basically use that to recognize when they're getting infected by another virus, right? And so how it actually works in humans or any other model organism that you want to use is it's a two-part system, right? So one part is the Cas9 protein. This is the protein that does the cutting and the modifying. The other part is your guide RNA. And this is what you design, right? So essentially all you have to do is have this PAM sequence nearby, which is recognized by the guide RNA as like a basic, this is where I should bind. But you design the sequence of the guide RNA and you design it to recognize a particular piece of DNA, your target gene. And so the aim is that this will only bind to that target gene and nowhere else. That's not always perfect, which, you know, is one of the downsides. But when you introduce these two things to your target cell, that might be human cell culture or inject it into C. elegans or into Drosophila. What happens is that this forms again, another scanning complex. So this is similar to the RISC complex in RNA silencing. But what happens is that this goes along and it finds its matching piece of DNA. And when that matching piece of DNA is found, Cas9 cuts it. It produces a double-stranded break. And so if that were viral DNA in a bacterium, then that would nix the virus, right? So that's kind of the original purpose. But here, what we're doing is that we're specifically cutting the genome at a very specific point. And that point is dictated by this guide RNA. Now, what happens now depends on whether or not there is any donor DNA available. So if there's no other DNA available, this double-stranded break gets repaired by a process called non-homologous end joining. And that's basically like, oh yeah, that free ends, let's stick them together, right? That looks about right. That'll be fine. It's kind of what the cellular machinery is saying. And almost always it introduces a mutation as a result, either duplication or deletion, because it's not a very good repair mechanism. It's kind of like a Hail Mary type response. Better to fix it than to have a break, basically, even if you don't fix it very well. Now, if in addition to these two things, right, the guide RNA and the Cas9 protein, now you add another bit of DNA, and that bit of DNA matches either side of that break. Now we'll use homologous recombination, which we learned about like way back when, right? Which is involved in crossing over in prophase, in cell division. If you provide a bit of DNA that matches that DNA, now your cellular machine is like, oh, phew, I've got another chromosome around I can use to do a better job. And any bit of DNA that is in between those matching bits now gets put into the genome. So if, for example, you wanted to introduce a green fluorescent protein gene, GFP gene, to look at gene expression, like endogenous gene expression, if you put that GFP sequence between bits of flanking DNA that match either side of that break, your cellular machinery will use homologous recombination to stick that in to fix that break. And so now you can modify the genome of your organism in a very, very specific way to generate what you want, right? And so the power of that is really, it's kind of unprecedented, to be honest. It now gives us the tool to do whatever we want with an organism's DNA. And that organism includes us, which is a little bit scary, to be honest. Anyway, unfortunately, that's brought us right to the end. Literally the end, like capital T, capital E. And I might even have to write that in the chat. The end. Oh, capital N as well. That wasn't meant to be there. Sorry. I guess it's fitting that we end on a bit of bad typing. But anyway, that's kind of it from me. It's been a fun ride. I've really liked teaching this section. It's been a lot of fun. Lots of good questions. Again, study hard. Exams on Thursday. Same format as before. Expect the same level of details. Ali, no, the only thing that will be on the exam is what we've covered today. Well, not what we've covered today, what we've covered in class up till today, right? So the biotechnology PowerPoint unfortunately didn't make the cut because we just didn't have enough time. Spent more time on the gene expression stuff. And so only the slides that I've talked about today from kind of here to here. The other stuff is just kind of information which I basically described in this one. And PowerPoints 17 and 18. So what slides is that in here? Sorry, come on. 11, there you go. So PowerPoints 17, 18, and slides 11 to, what's the last one? 21. Of PowerPoint 20. That's all that will be on the exam. That's not all, because obviously there's an awful amount of information there, but any of the stuff in the rest of this PowerPoint or in PowerPoint 19 will not be on the exam because we didn't cover it in class. All right, I know some of you have other places to go to, so it's been a pleasure. I'll still see some of you in lab tomorrow, and I'll be around for the at least first half hour of class on Thursday if you have any questions about the exam. So good luck, have fun, and I'll see you around, hopefully.
Mr. Crook, I had a quick wanted to let you know real quick. I have you for lab tomorrow. And I might be 5-10 minutes late. I have to take my little girl to the doctor in the morning, and her appointment's at 9:50. So they're usually really good about being like 30 minutes, but sometimes they're like, we're there for an hour. And it's on the opposite side of town.
Oh, yeah, I know, that really sucks.
Yeah, she's sick. I didn't get any sleep at all last night.
Oh, I'm sorry. I hope she's feeling better after she gets to the doctor at least. Oh, is that
hopefully, but I just wanted to let you know.
Oh, that's your dog. So that's fine. Yeah. Just read the lab handout ahead of time if you can. It will give you a bit of a jump, and get there when you can. And we'll cover as much as we can. It's kind of like a self-guided lab, and so coming in a few minutes late isn't going to really affect much.
Okay, cool. Just thank you so much.
Yeah, no worries. I'll see you tomorrow. All right. Catch you later, everybody.